All right, let's move on to talk about our benign breast tumors. We're going to start out by talking about fibroadenoma, and this is going to be common in our females under 35, and this is actually going to be the most common breast tumor in our patients under 35, and this is really reassuring because this is a tumor that has no increased cancer risk. So if a patient has this, there is no increased rate of cancer. This is going to be completely benign, and importantly on exam, this is going to be a small, mobile, well-defined mass, and this is a little bit different from our fibrocystic changes, which are a little bit like homogeneous, kind of these fibrous threads or cords that can be present bilaterally, whereas our fibroadenomas are going to be a single discrete mass, and this is going to be composed of fibrous tissues and glands, and these are going to be hormone sensitive. So as during the menstrual cycle, when a patient has increased estrogen, we're going to get increased size and tenderness of this tumor. So a patient during a certain part of their cycle may complain of increased pain or notice that this part of their tumor gets a little bit bigger. And if we look at this on ultrasound, we're going to see here that this is a really discrete, well-defined mass. And I want you guys to take note of the color of this ultrasound. So the tissue here really matches the surrounding tissue because this is kind of a fibrous, fatty tissue that matches the surrounding breast tissue. And this is going to be in distinct contrast to our next tumor, which we're going to talk about. So let's move on now to talk about our intraductal papillomas, which I'm going to highlight here in blue. And these are going to have a small increased risk of cancer. And these are going to be located in our ductal tissue. So if we zoom in, our ductal tissue is right here underneath the areola. That's where all the milk that comes from the glandular tissue is going to coalesce. And with these tumors, we're going to see fibroepithelial tissue within our lactiferous ducts. And like I said, this is going to be classically subareolar. And these can have serous or bloody discharge. And this is going to be a key part of history. If you have a patient with some discharge and a lump, this is going to be on your differential. And I want to point you out towards the ultrasound findings here. And notice that this tumor here is going to be composed of fluid. So it's a really fluid-laden tumor, and fluid on ultrasound is going to be much more black than a fibroma. So if you look, the tissue characteristic here is going to be different. This matches surrounding tissue, whereas this is much blacker than the surrounding white tissue here, and that's going to be a key thing to differentiate these two types of tumors. Lastly, let's talk about our phylloides tumors, which are going to be really high yield on the exam because they really stick out, and this is going to be common in our older patients. So this is going to be ages 50 to 60. There's a small portion of these tumors that are going to progress to malignancy, but this is usually a small risk, and these are usually going to be a painless tumor. These are going to present as a large mass, and this is all about histology here. So the mass is made of connective tissue and cysts, and if we're to look at this under our histology, this is going to be described as leaf-like lobulations. This is super high yield. If you look here, these look like leaves. If we were to kind of like outline them, these are like leafy projections. This is a histology picture you guys really want to burn into your memory because it's super high yield. If you see this is going to be a phylloides tumor until proven otherwise. If you hear that buzzword leaf-like lobulations, this needs to be on the top of your differential. Let's move on now to talk about our non-invasive breast carcinomas. So these are going to be carcinomas which are not going to be completely benign. However, these are tumors that are non-invasive or not in an invasive stage of their presentation. Let's start by talking about ductal carcinoma in situ. So within the name, this is going to be all about ductal tissue. So we're going to expect it here in the area of the ducts as opposed to the glands, and these are going to be characterized by ductal atypia. So we're going to have a lot of cancerous cells that are going to be filling the ducts of the breast tissue. So our ductal lumens are going to be chock full of tumor cells here, and these are going to be characterized by microcalcifications on mammography. And importantly here, this is an in situ lesion, and in situ means it has not penetrated the basement membrane. So there's going to be no basement membrane penetration here, and there's actually going to be a special subtype that might come up on test day called cometocarcinoma, and this is going to be characterized by high-grade nuclei with central necrosis as well as dystrophic calcification. That dystrophic calcification might be something that could be picked up on mammography. And let's take a look at this histology here. If we zoom in, we're going to see that this is all cancerous material filling the duct lumen. So the duct lumen I have kind of outlined, and all this cancerous material is in here. We're going to see there's like a lot of nucleic activity, right? There's a lot of nuclei in here, and these are all cancer cells, but this has not yet penetrated the basement membrane, so this is still considered an in situ lesion, and so this is going to be ductal carcinoma in situ, has not pierced through the basement membrane. Let's move on now to talk about Paget's disease, which is going to be another form of ductal carcinoma. However, this has like spread to the epidermis. So remember previously how I was talking about how ductal carcinoma happens right here at the duct? Our Paget's disease of the breast is actually going to occur out here on the skin. And so this is going to be an important differentiation. It's the same pathology happening here, but it's going to be a different location. So we're going to have eczematous patches on the nipple and areola. And an important thing on your differential here is going to be eczema, because these lesions look just like eczema. And so like I said before, this is going to be a DCIS, extension to the skin of the nipple, and it's going to be characterized by Paget cells. And Paget cells are going to be intraepithelial adenocarcinoma cells. A great example of it is the histology that I have here. So if we zoom in and take a look, this is beautiful, beautiful histology. We also have Paget's disease in other parts of the body, and it always looks like this. So we have this really beautiful hyperkeratotic lesion here at the top, and then we have like these vacuated cells, which are characterized as Paget cells, and you can see they're all throughout here in the epidermis, and then we have our underlying dermis below, and it makes these beautiful layering effect, and this is going to be characteristic of all Paget's disease across the body when it comes to skin. And if we were to look at this on physical exam, it would look like this. It's like this really eczematous area around the nipple, and this is going to be classic for Paget's disease of the breast. Let's wrap up by talking about our lobular carcinoma in situ. So this is going to be disease process of the lobules of our breast. So previously we were talking about nipple and ductal, but now we're talking about our lobule area, which is going to be all back here. This is going to be our milk producing tissue, and this is going to be something that's found incidentally on biopsy. And so when we find it incidentally on biopsy, then it's going to alert us that this patient needs to be monitored. Although this is an in situ lesion, it has the potential to progress to not be an in situ lesion anymore, aka to pierce through the basement membrane. This is going to be an increased risk of cancer in either breast. I want to zoom in here and talk a little bit more about the molecular biology of our lobular carcinoma in situ. So I'm going to draw two layers of connective tissue for us here, and then I'm going to insert these molecules called cadherins, and I'm going to designate them as these little boxes here. And cadherins, if you remember from the molecular biology importance on step 1, are going to be connective molecules, and they're going to be calcium mediated. So I'm going to draw in these little calcium blue dots. I'm going to label them here Ca2+, because that is how we denote calcium in our step 1 exam. And then there's one more player here, and that's going to be catenins. And catenins are going to help with the function of cadherins. So I'm going to draw these green dots kind of lacing through this matrix here, and these are going to represent our catenins. And these are all responsible for a connective tissue function. And so if you have a lobular carcinoma in situ, we expect them to have decreased cadherin expression and to be P120 catenin positive. And this is essentially like the initial brewing factor of this type of cancer. And this type of cancer can progress to not being in situ anymore. And when this happens, we're going to get some changes that I want you guys to hang on to, because we're going to talk about the non-in situ version of this cancer. But I want you guys to note the important molecular players when talking about this type of cancer. And so if we look here, our lobular carcinoma in situ is going to be here, and this is because this is all confined within the basement membrane. And this is as opposed to our invasive lobular carcinoma, which I'm going to highlight in red here, which has kind of escaped the confines of the basement membrane. And we're going to talk about this a little bit more in the next slide, but I wanted to kind of prime you guys for that. So remember, these are all going to be non-invasive carcinomas on this slide. They have not broken through the basement membrane, and we're going to move on now to our invasive carcinomas in the next slide.